Check engine light on? Take the guesswork out of your check engine light with O'Reilly Veriscan. It's free. Ask for O'Reilly Veriscan today.
Auto Parts.
Pretty thighs, shawty, she a dime. Demon girl, evil eyes, she be telling lies.
All right, so today it's my pleasure to interview Dr. Schaffer, co-founder of 40 Molecular Therapeutics and professor at Berkeley. So Dr. Schaffer, thanks for being part of this episode. So can you tell us how you got started and
Sure.
your background?
My background is that I'm a part engineer, part neuroscientist. But going back a little bit further, probably how I got started is that my mother worked in late-stage drug development, was an MD who ran clinical trials at Sanofi and Novartis, and my dad was a very early-stage basic biochemist, was a professor in academia. And parts of what they both did appealed to me, so I basically landed halfway in between, where I'm very happy to be a professor, to have the freedom to be able to work on problems that are curious for us or interest us. But I'm also very interested in getting the products of that research into human clinical development to try to have a positive impact on human health. So I've really landed at that interface of being a professor, but also playing a role in transitioning the technologies into startup companies so that we can try to help, human health.
Yeah, I mean, I just want to ask, it's really interesting, of course, both parents working in the biotech industry and other stuff. I think previous cats, we had Bob Langer, the Moderna co-founder, and his son was on here. Also the family was a biotech family. So I'm going to ask, did your parents, kind of persuade you to join the biotech field, or do you kind of choose on your own?
I chose on my own.
Okay.
They were very open-minded. I'm a father now, and I'm very supportive of my kids doing anything where they could make a comfortable living. So my parents were pretty similar, but just happened to gravitate towards, landing somewhere kind of halfway in between what their own career interests were.
Yeah, so let's talk about your research. Of course, I know you're in gene therapy and stem cells. So what got you interested in gene therapy specifically?
When I went to graduate school, I was looking into two different research groups working on two different therapeutic modalities. One was gene therapy and the other was antibodies. And in both cases, the goal was to engineer them to turn them into better therapeutics, using principles of chemical engineering. You mentioned Bob Langer. He was actually on my thesis committee during my PhD. I'm a chemical engineer by training. So very, very fond and obviously have a huge amount of admiration for Bob. So I was considering antibody engineering and gene therapy. At that time, there were FDA-approved antibodies already. And one of my self-acknowledged kind of personality traits is that I tend to like calculated risk. I like swinging for the fences. I'm not going to do it. I'm not going to step off a cliff, stupidly. But I like looking at places where if I can think it through carefully, I can identify ways in which I can take a risk and then hopefully, if things work out, have an impact. So antibodies, like I mentioned, there were a number that were already FDA approved. Gene therapy was blue sky, so that's the direction I went.
Mm-hmm. Yeah, so for your research regarding gene therapy, can you talk more about how you're going to use viral vectors to enhance the ability to deliver genes or drugs?
Yeah, I mean, the underlying concept of gene therapy is that the therapeutic product is a piece of DNA. So you're trying to treat disease by delivering a gene, delivering DNA. And there are a number of ways in which that could be potentially transformational. You could have really a long-term therapeutic benefit if the delivery efficiency is high enough, and you could treat the underlying cause of the disease down to the genetic level, which can further have a potentially a pretty broad effect on efficacy and potency. The big problem is, the field has this very old joke that there are only three challenges in gene delivery or gene therapy: delivery, delivery, and delivery. And, getting a piece of DNA into enough cells to have a therapeutic impact was a really tough thing to do. So viruses do that as part of their life cycle. They carry nucleic acid inside of cells. And so we work with DNA viruses, which carry DNA inside of cells. And the goal is to find a virus that's pretty safe, has some reasonable starting point and efficiency, and then engineer that virus to be much more efficient and in some cases targeted, so that you could cross that threshold of being able to have a positive impact within a patient by delivering enough of your medicine, your piece of DNA, to the cells of that patient.
Yeah, I mean, so no, on this podcast, we've had some CRISPR guys on, CRISPR people on there. No, example, George Church was on there. So does your gene therapy research also incorporate CRISPR stuff, or is it just the whole the chemical side of it? There's no biology involved. Like, do you guys also do like CRISPR research as well?
Yeah, I mean, the a gene therapy is the combination of the delivery vehicle and the therapeutic cargo. Often people have kind of focused their companies or their careers on one side or the other. That's not always the case, but in some cases people are leaning a bit trying to solve the delivery problem. In other cases, people are working on kind of next generation enhanced cargoes. So CRISPR, Cas9 is a cargo. It needs delivery. So we have within my lab, my academic lab at least, delivered Cas9 to be able to treat a number of different conditions. We work on engineering improved technologies based on Cas9. But by and large, my lab leans a bit more towards delivery.
But do you guys collaborate? Like, I know Dr. Jennifer Dodona is also in Berkeley. Do you collaborate with her as well?
Yeah, Jennifer Doudna is, phenomenally talented and accomplished scientist. Obviously, being awarded the Nobel Prize several years ago for her first discovery of how to engineer CRISPR to retarget it towards novel loci in the genome. So I have published with Jennifer. We worked on a delivery actually. So we were interested in developing efficient technologies for genome editing cells and culture. And so we combined together her ribonuclear protein complexes, her Cas9, with a piece of DNA delivered by AAV, which served as the donor for the genome editing at the target site.
Yeah, so I'm gonna talk about more towards the public now. So I mean, of course, during the COVID-19, people were fearing that the vaccine made by Moderna was some kind of gene therapy and stuff. So I want to ask, like, what do you have to say regarding this concern that gene therapy could have potential unknown long-term side effects on people's genes? Like maybe there could be some like errors where like the therapy may have, done some alterations in someone's gene by accident. Do you think that's going to be a concern? And how do you think we're going to solve that? No.
Sure. Just to take a step back,
Yeah.
so the Moderna and the Pfizer BioNTech vaccines, those were messenger RNA,
That's yeah.
yeah, delivered by lipid nanoparticles. So messenger RNA is a transient molecule inside of a cell. So it lives long enough to express to produce some protein, and then the goal was for the immune system to recognize that protein as foreign, that COVID or SARS-CoV-2 protein, recognize it as foreign and mount an immune response against it. And the memory of that immune response is the basis for the vaccine. But once the messenger RNA has produced a bit of the SARS-CoV-2 envelope protein, the messenger RNA gets degraded and goes away. So there's no long-term effect associated with the messenger RNA altering the cell. It was just used as a way to stimulate an immune response. And we all, hopefully, all of us have a long-standing memory of vaccines. I got the polio vaccine, and you can kind of go through the long list of tetanus, pertussis, diphtheria, et cetera, et cetera. So we've all had lots of vaccines where the outcome was the same, that we ended up with immunological memory against the antigens that were delivered, so that we can mount a quick immune response against the pathogen if we ever unfortunately encounter it. But the vaccines themselves go away. They get degraded and cleared by the body. Same thing with the Pfizer, BioNTech, and the Moderna vaccines. Those just get cleared out by the body, and the thing that remains is our immune system being primed to go after the pathogen if we ever get infected with it. So there's no permanent effect of the messenger RNA altering a cell's genome. Gene therapies, if you're delivering a genome edit, the goal is to alter the genome, right?
Mhm.
but to do so in a targeted, intentional way so that you can fix a broken gene or otherwise repair the cell for the impact of a disease. So in that case, the goal is to have a long-term effect and for that effect to be therapeutic.
Mhm. Yeah, so I know 2023, the FDA, they approved first two gene therapies. It was Casgevy and Lyfgenia for sickle cell disease. So I'm just, asking your opinion. Do you think like the government, the FDA, are they starting to have more confidence around gene therapy? As I know previously, like they weren't too sure if they wanted to approve it or not. Like, do you think the governments are starting to have more confidence in gene therapy and are pushing it to make it more accessible to more people?
Yeah, there are. So I would define a gene therapy as the delivery of, therapeutic DNA to be able to treat a disease. And in some cases, that overlaps with genome editing, where the goal is to deliver an enzyme, and in some cases, some donor DNA to uh, result in a targeted change in the sequence of a gene inside of our genome. So, to the extent they both involve delivery of an exogenous piece of DNA, they're both gene therapies, but they're kind of, overlapping in a Venn diagram, but not identical to each other. Um, so gene therapy, they're actually on the order of 20 FDA-approved gene therapies. The
Oh,
first one was approved in 2015, and they've been approved for a variety of things for rare monogenic disease, using a couple of different vehicles. They've been approved for cancer in some cases, B-cell lymphomas and leukemias. Casgevy, or the genome edits you mentioned for sickle cell, those again, it has some strong similarities to gene therapy, but they were kind of Casgevy was the first genome edit that was approved by genome editing technology approved by the FDA to treat a condition. And I think that whether it's a gene therapy, quote unquote, traditional gene therapy, or a genome edit like Casgevy, the FDA is, I think, gaining increasing comfort with providing regulatory approvals to those based on their safety and efficacy records in human clinical trials.
yeah. So I'm going to talk about like the political dynamics now. And of course, I know you co-founded, I think, a couple companies. One is on NASDAQ. So I think with the incoming Trump kind of administration, I know biotech is in this position where it's uncertain if they're going to get their drugs approved faster or slower. I'm asking your opinion. Do you think, you know, this year and maybe in these 4 years, a lot of therapies will be approved faster, or do you think there are just going to be more rules?
It's so I've co-founded 7 companies based on research out of my lab.
Yeah.
I've either taken them public, they've been acquired, or 4 of them are private and everywhere from Series B to pre-seed right now. So I've had, a lot of experience in contact with the capital markets and the regulatory agencies. At this stage, it's about speculation. We all
Yeah.
have things that we're concerned about. For example, last week, the federal government placed a temporary hold on all external communications by the NIH and the FDA. People are speculating, but are not entirely sure why or what the rationale is for that. Obviously, if communications are held for a while, that's going to have a pretty significant impact
Mhm.
on the ability of companies to advance their programs into the clinic, through the clinic, and towards regulatory approval. So not sure how long term that is going to be. When it comes to the capital markets, a tariff is a tax, right? Consumers have to
Mhm.
pay more for a product that comes from overseas. Uh, an increase in the cost of a product is inflationary. Inflation has been a problem within our economy
Mhm.
for the past several years, and the Fed has had to hold the interest rates high to be able to combat inflation. If these tariffs end up being, continuing that trend of inflation, interest rates are going to stay high for a while, which actually serves as a headwind for investment in biotech because it's safer to just stick your money in the bank and earn a higher interest rate than to invest in biotech. So that's going to be, challenging for biotech from both the private as well as the public markets. So, but it all comes down to speculation. We're not exactly sure how this is going to develop, right?
I think it's going to hurt the startups even more. I know you mentioned you have some startups. because I know they're like, it's not like there's some big company with like unlimited cash, right, coming in. Like they're probably not even profitable yet, and like like these rules come in, so I think it's gonna hurt the startups even more, actually.
I think it's going to make it harder to get access to capital. Yeah.
Yeah. Okay. So my next point is, I know you also do stem cell research. So do you think stem cell therapies could help with like Alzheimer's?
There are, so the reason that we're interested in both gene therapies and cell therapies is that there are a number of kind of long-term progressive degenerative disorders where, cells gradually die away in a particular location of a tissue or even the whole tissue in some cases. And, if you catch it early enough, if you can deliver a piece of medicinal DNA as a gene therapy, you could potentially halt the disease or perhaps even reverse. If the cells have died off too much, then you really need to replace function. And in some cases, that means you have to replace the cells. So in the first case, you're talking gene therapy, and in the second case, it's a cell therapy, and they complement each other in a number of ways. So as a couple of examples, Parkinson's disease is a condition where typically by the time somebody is diagnosed, they've lost 50 to 80% of the affected cells, the midbrain dopamine neurons within the region that's affected in the brain. And so if you catch that early, perhaps you could deliver a rescue to halt the degeneration of those cells. If you catch it late, we're thinking about a cell therapy to replace those cells. Type 1 diabetes is caused by an immune response against somebody's own beta cells, which are the insulin producing cell inside the pancreas. Perhaps if you catch it early enough, which is a little bit hard to do, you could try to induce tolerance with the gene therapy and halt the progression. If you catch it too late, then you're talking about a beta cell replacement therapy. So I really view those as complementary to one another.
Mhm. Yeah. So I know many athletes think Aaron Rodgers, they use stem cell therapies to heal injuries. I mean, I do you think it's possible for stem cell therapies to potentially be like the main thing that speeds up the recovery of many of these athletes who want to get back to the field?
Yeah,
Like, do you think?
it's very speculative right now. What a lot of these involve is, there are a number of different types of stem cells. It, it became somewhat quote unquote popular to take bone marrow stem cells, and, isolate, do a little bit of a bone marrow tap, expand the cells, and then put them back into the body to try to treat tissues that are suffering from some kind of injury or degeneration. While it's a kind of popularized idea, and there are lots of clinics that are doing it, some within the US, lots overseas, it's really, I think, sketchy whether there's a therapeutic benefit. So personally, as somebody who's worked with stem cells for 27 years, I wouldn't recommend doing that because the documented therapeutic benefits are not well justified yet, and there are risks associated with it.
Yeah, I don't know. I think especially in football, I don't know. You read it's not just Aaron Rodgers. There's a couple other football players who have tried this and they said, "Oh, it works. It's so good." It actually shortens my time. So that's why I asked the question, because I know it's getting
Yeah,
popular.
dating back into Peyton Manning, I think, who also, did injections into, I think, his neck or something like that, if memory serves. I mean, really the way that you prove something is you do a statistical study, and you have it's a controlled study where you have at least two groups of patients, one who receives the therapeutic, the other one who receives something that makes them think like they've gotten the therapeutic. So you get to compare the efficacy of the actual therapeutic to the placebo effect of the people who didn't get treated with it. And if you do that in large enough numbers, you can do the statistics. You can say there was a benefit or there wasn't. And if there was a benefit, here's how big it was. And that would be the kind of study that would need to be done to, I think, really strongly recommend that as a therapeutic for a sports injury.
Yeah, is this similar to stem cell tourism? So basically, what it is like there are countries like China and South America,
Mexico.
and yeah, Mexico. They have these what's called stem cell treatments, and people go there. Do you think these are scams, or do you think these could be maybe something that could work for the public? Because it feels like to me it's like sketchy.
I think they're scams, and I'd recommend against them. Or at the very least, if somebody got into contact with one of these clinics, they should, if they're taking a risk by going to the clinic, they should demand to see the clinical data from the previously treated patients showing, really documenting carefully the therapeutic benefit. And it shouldn't just be one or two patients who anecdotally say, hey, my knee feels better. It should be, a couple dozen patients where there's documented MRI data or similar showing an improvement or a recovery of function. But still, I think that places that have a strong track record for doing, placebo-controlled blinded human clinical trials are the ones that are most likely to generate strong clinical data to prove or disprove the efficacy of a novel therapeutic. This could be in the US and Europe and Japan and Korea. There are a number of places that in Australia, have a track record of running high quality clinical trials. And so I wouldn't resort to a clinic within a country that's not compatible with that.
Yeah, so I think another argument is like the healthcare costs. So regarding like gene therapy and stem cell treatments, what do you think are some way to lower the cost? Do you think getting rid of like the middleman between, I think you probably heard about it's like there's the biotech and there's a pharmacy and there's some middle manager guy in the middle who does all the transactions. Do you think getting rid of them would make the cost lower?
Yeah, kind of like an HMO. But I mean, in the case of gene therapy, the costs are high for two reasons. The first is the nature of the disease being treated. In most cases, the disease is a rare disease. It's hemophilia, it's muscular dystrophy, and drugs for those diseases have always been high in cost, regardless of whether or not it's a gene therapy. And the reason for that is that the government recognized that drug companies were not developing therapeutics for those diseases because it wasn't a large enough market. The company was not going to get enough reimbursement and enough upside for taking the risk of developing a drug for that target. So the government passed the Orphan Drug Act to incentivize companies to develop therapeutics in those areas. And part of the outcome of the Orphan Drug Act is the cost of the therapeutic is going to be higher, because if you're not treating as many patients in order to be able to get a return on the big investment that went into the company to develop that therapeutic, you need to charge more per patient. So that was a direct outcome of the Orphan Drug Act. Some people say that's gone a little too far and prices have gone up too much, but that was initiated by the Orphan Drug Act. And the second reason is that gene therapies ideally are a one and done. So most companies, if they've developed a therapeutic, whether it's a statin or
Ozempic.
or Ozempic or recombinant protein to treat hemophilia, the effect of those drugs are transient. So a patient has to take their hemophilia drug, their protein, the rest of their life. With the gene therapy, it's one and done. So they don't get paid every year for the rest of a patient's life. So they have to, recoup a certain amount of a payment up front, or maybe over the first 10 years after a patient's received the therapeutic, to recognize the fact that they're no longer going to be paid by the insurance company or the patient 10, 20, 30 years from now. So as an example of that, there was a recent FDA approval for hemophilia B by a company called Unicure. And the previous drug for hemophilia B was a recombinant protein therapeutic. It works well. There's still some bleed throughs. Patients still need to go to the ER. But that recombinant protein is pretty expensive. It's on the order of $150K a year. Patients, thankfully, are living a normal lifespan. So if you do the math and add up everything, the total patient costs of that therapeutic are greater than 20 million for the total lifetime of the patient. And Unicure is charging, I believe, around 3 million for their therapeutic. I should look that up. It's between 2 and 3, I believe. So you could say that gene therapy, even though the sticker price may raise eyebrows, it's actually saving on the total lifetime of treating that patient for hemophilia.
Yeah, I mean, now why I brought this up was you heard of Mark Cuban's Cost Plus Drugs pharmacy startup?
I'm not. I've heard of it. I have to confess, I'm not familiar with the
Okay,
business.
all right. Yeah, so that was why, because they were talking about doing like a wholesale deal instead of going through some middleman person,
Mhm.
basically. All right, so let's go to your companies. I know you co-founded 4D Molecular Therapeutics. It's also on the Nasdaq. So can you explain what 4D does?
Sure. Again, gene therapy comes down to delivery, delivery, delivery. And back around 27 years ago, I started working with a particular virus called adeno-associated virus, or AAV, which had a number of characteristics that made it look like it could possibly be an interesting gene therapy virus. Viruses carry DNA inside of cells as part of their replication cycle. A gene therapy requires delivery of DNA, so there's been a longstanding interest in taking viruses and effectively tricking them into carrying a therapeutic medicinal piece of DNA rather than delivering and carrying in their own genes. And AAV looked like an interesting candidate for this approach. Natural versions of AAV have not been engineered as therapeutic gene delivery vehicles, as pharmaceuticals. They have a set of properties that have been shaped by natural evolution, and they're not optimal for delivery to most tissues inside the body. And we developed this idea of taking a technology that was originally invented for enzyme engineering, called directed evolution. And in fact, Norbert was awarded the Nobel Prize in Chemistry in 2018, for incredible outcomes in enzyme engineering that were enabled by that technology. So that technology began to emerge in the early 1990s. I started my lab in the late 1990s, and so we took directed evolution and we're the first to apply it towards AAV, towards creating engineered, improved versions of AAV that were much more efficient and in some cases targeted compared to the natural versions of AAV. At some point, I became very interested in getting that into human clinical development, so that we could see whether or not it could help patients. And vectors, versions of AAV that we've created have gone into on the order of 10 human clinical trials. A number of them, several of them run by 4D.
Mhm. So I know these viral vectors, they can cause like immune responses to human beings. So have you explored like non-viral vectors in
I
these gene
worked,
therapies?
yeah, I mean, I worked with non-viral vectors for my PhD. Um, I think viral vectors have the ability to be toxic. Non-viral vectors have the ability to be toxic. Both need to be engineered to have pretty inert interactions with the immune system. There have been a number of immune responses in AAV gene therapy clinical trials, particularly the ones that have involved injections of very large doses of, first generation AAVs, natural versions of AAVs, into the bloodstream. That's where the majority of these immune responses have happened. So if you can take an engineered virus where you can drop the dose 10 or 100 fold, that ends up getting, but still have, the same or even better delivery, ideally better delivery efficiency, that ends up taking the virus down below the radar of the immune system, so that ideally the human immune system is not going to notice it. So I think there are multiple paths towards solving this problem of immune responses against nucleic acid delivery and gene delivery.
Yeah, so no, I took a look at your company. So it looks like 40 stocks has been falling since it IPO'd around 2020. So I want to ask, like, what do you think there's some way to make sure the 40 stock goes back up? Like, do you think you need more time for your therapies to go through trials?
Yeah, I think that some of that is that biotech has come down a lot.
It's true.
And some of it is that, I think there were some questions about the company's therapeutic pipeline, that the street liked some disease targets, didn't like other disease targets. So I think there was, some of the choices that were made by management were not well received by the street. So there are a couple of products that are viewed as the most promising. And for those to get into a pivotal clinical trial, meaning a clinical trial that would generate data that could lead to regulatory approval by the FDA, for those to get into a pivotal clinical trial and to execute and follow through on the promise that's been seen in the earlier clinical trials and get to an FDA regulatory approval, getting onto that path is what leads, for any company would lead to recovery in the stock price.
Mhm. Yeah, so I see 4D has partnership with some big pharmaceutical companies. I think you guys had one with Pfizer. So how hard is it for, you know, especially for startups? Not talking 4D, like for startups, you get like a partnership with a big pharma company.
Yeah, it's challenging. I'll take one step back and say that I have not been affiliated with 4D for several years now, so I don't
Okay.
consult. I'm no longer on the board of directors. At some stage, the company, as it should, transitioned into being a clinical operations company. And I learned a bit about clinical operations and clinical trial management, but I'm not an MD, I'm a PhD. So I ended up pulling back in parts so I could pivot my bandwidth to some earlier stage technologies to try to get them into human clinical trials. But speaking to your question about partnerships, I, you know, the strategics are looking for technologies and therapeutic areas that are complementary, that are strategic for their own internal programs, are looking for differentiation, a potential smaller or larger partner company that has a technology that is differentiated from the rest of the field and has the lowest possible risk of ideally of succeeding for a given direction for a given clinical target. And they're looking for management that is capable of executing on that, and in some cases, ideally, a track record of success. So all in all, yeah, not the easiest thing to get a pharma partnership.
Mhm. So you co-founded Ignite Immunotherapies that was acquired by Pfizer. So, I've had some VCs on here and they talked about that like the immunotherapy area is such a hot field right now. So, I mean, why do you think like especially immunotherapies is generating so much interest in the VC community and also from biotechs?
Yeah, it's that there have been multiple generations of therapeutics to try to treat cancer, right? And within the field, there started emerging. Actually, this was a concept that was really invented at UC Berkeley at my university by a colleague of mine, Jim Allison, who's since moved on to MD Anderson. But Jim started proposing this idea, a new concept for treating cancer. And the idea is that cancers are obviously non-self. They start behaving in a aberrant, abnormal way, and start behaving in a way that is harmful to the rest of the tissues in the body. So in a sense, they're non-self. Immunologists would say there's some aspects of a cancer that's non-self, including the fact they acquire mutations and acquire changes in protein sequence. Um, so he hypothesized that the immune system, whose job it is to discriminate between self and non-self, should have the ability to recognize a tumor. And it turns out, the field as a whole has discovered since then that cancers have a wide, pretty broad range of strategies to actually suppress host immune responses against the tumor. So he proposed that if you actually started kind of turning off the ability of the cancer to suppress the host immune response, you could really rev up the host immune system and empower it with the ability to attack the tumor. So that's cancer immunotherapy. We know the immune system is incredibly powerful at fighting pathogens, and the strategy here was empower the immune system to attack a tumor. And that's led to several classes of cancer immunotherapies, both monoclonal antibodies as well as cell therapies. And those have succeeded against, a large number of malignancies. I think it's fair to say there's been a bit of a slowing down in the field because some more recent clinical efforts haven't always panned out as well as some of the first generations did. But there's still a huge amount of hope that we can find new mechanisms and new ways to arm the immune system or empower the immune system to go after the cancers that have so far been refractory to cancer immunotherapy.
Mhm. Yeah, so our next question. So I think I read an article recently. They're saying that a lot of US biotech and especially large pharmaceuticals have been uh licensing some experimental drugs from Chinese biotech companies. And then so people are now worried that the US biotech industry could lose its edge over Chinese companies. So I mean, I'm just gonna ask you, so, what do you think are some ways to make sure the US biotech industry can remain very competitive?
Well, I think the US biotech industry is highly competitive right now. I think that it's between Boston and San Francisco and San Diego and Seattle. there's a huge amount of innovation, of new technologies that are being developed. And what enabled that was a combination of very special ingredients in the U.S. Extremely educated workforce, significant investment from the government in the form of the NIH and other entities that invest in early stage biology and early stage technologies that then translate into therapeutic products. A investor community that is risk friendly, that's willing to invest in early stage technologies and get them to the point where they're close to regulatory approval. And then we also have a strong pharma presence that's capable of taking those technologies and effectively and efficiently commercializing them and getting them into the clinic and distributed well. And then there are other components of this, incredible university system, incredible academic hospital system. So we're very competitive. We're not foolproof, right? You can break some elements of that system if bad policies start emerging, but we are very competitive right now. And in some cases, the vast majority of cases, I would say, the early stage technologies are emerging from the US. On occasion, they're coming from Europe. On occasion, they're coming from China, where the US decides to license those technologies and develop them into therapeutics. But the US is still the leader when it comes to biomedical innovation and biomedical research for the moment.
So I know more in the article, but so basically what the point is, what they're saying is like the Chinese biotech companies can move faster. So let's say like you have a startup, right? They can go from their launch to clinical trials in 18 months or less. So that's that, which is better than a few years in the US. So, do you think there's some regulations that need to be cut? Like of course I know it's important for the public. Do you think there's some regulations that just the government needs to get rid of to make it more efficient and faster? Or do you think for now it's fine?
I guess it depends on how you measure the time. Is it from the point where you discover a clinical candidate that you want to advance into the clinic to the point where a phase 1 clinical trial is initiated and then to when a regulatory approval happens? Or is it from the point where there's some decision to invest in a basic discovery and it gets all the way to a new technology and then to a clinical candidate? I would say overall, the US is still quite efficient. Maybe during pieces of that, like how do you get from a clinical candidate to a clinical trial being initiated? That process is tightly regulated in the US with the intention of maximizing safety to patients, whether it be the manufacturing side of it, the GMP manufacturing, the preclinical studies in animals to show safety and efficacy, the FDA review process, that those are tightly regulated. And I think the FDA, perhaps at various times of its history, has kind of swayed a little bit more towards let's be fast so we can get new therapeutics into human patients as quickly as possible, to, something bad happened in these clinical trials. We need to be more careful and, value the welfare of patients who are, entering into these phase 1 clinical trials more. So that's kind of a moving target that evolves over time. But, I would say that, putting a halt on FDA external communications with the rest of the US public or with the rest of the world is probably not a good step towards making human clinical trials more efficient.
Mm-hmm. Yeah, so I know you're the director of the Bakar Labs. So can you explain more on what it is? Is it like a life science incubator or is it like a venture capitalist firm?
It's a bit of both. So the Bakar Labs are currently a single incubator going on a network of incubators. We're 92,000 square feet of space, which makes this the largest university-owned and operated biotech incubator in the country on the UC Berkeley campus. But we're adding on a UCSF incubator in biotech. We're adding on a climate tech incubator on the Berkeley campus as well. So we're branching into other areas of impact, and we're working on other projects as well. But the overall goal is, our motto is entrepreneurship for the public good. That there's lots of innovation that happens within universities and has a hard time making it past the elegant publication and into the private sector because technologies need to make it into a company in order to scale to a solution for a problem that humanity is facing. So our goal is to lower barriers to technology translation from academia into companies, and as a result, accelerate the process of turning technologies into solutions to societal problems. We do this with a variety of parts of our program. We fund late-stage academic research. We work closely with venture capital funds and help set up a VC fund. We have our wonderful incubator, state-of-the-art laboratory space, and like I mentioned, we're going on several incubators. We have a network of larger partners from pharma, biotech, banking, legal, real estate, equipment, where we help our small companies network with those larger entities, as well as network with investors to get access to the resources, the knowledge, and the capital to accelerate the technologies. And so Bakar Labs has been, so the incubator has been open. We call the incubator BBH. That's the name of the building. So that's been open now for about 3 years. We just celebrated our 3rd anniversary last November. We have been home to so far over 40 companies, around 43 companies, which are current companies, 29, as well as 14 alumni companies. And during their time with us, the companies have collectively raised over $600 million and created over 400 jobs in California. So we're very proud of how we're impacting, I think, innovation, entrepreneurship, biotech, as well as stimulating the California economy while we're at it. So we're very proud of that, and very proud that we have the opportunity to now extend that success into another major human problem, or a problem for our planet, which of course is climate change.
Mm-hmm. Yeah, so I've talked to other scientists who are like CEOs of their own like research institutes. So I'll give you some example. Buck and Terasaki, they're all in California as well. So what makes Bakar Labs different from these other incubators? Because I know they have their own stuff going on as well.
Yeah, it's an institute up in Novato that focuses on human aging. And so I have a lot of respect for the Buck. Their focus is primarily on sort of non-company, more basic research. And that's not to say that the Buck will create a technology that will get spun out into a company in some cases, but really the focus of the Buck as a whole is to incubate basic research. So Bakar Labs is an institute that is focused on translation of technology from an academic institution into a company. I'll mention as well that we actually serve the needs of companies not only coming out of the UC system but also elsewhere. We're looking for highly impactful technologies and talented teams who are working on important problems.
Yeah, so I see there's the application form for Bakar Labs. So I was wondering what the acceptance rate of these companies that actually having a space in the Bakar's lab is, and like what factors do you guys usually take notice in some startups that have gotten these spaces?
Yeah, we're looking for several things. One is the problem, the societal need that is being addressed. There has to be a strong societal need that the company is focused on, whether it be, cancer or heart disease or infectious disease. We're looking for a really differentiated, innovative technology. And we're looking for a, very talented and seasoned team that ideally has a track record of making progress and translating technologies in the private sector previously. So those are really the ingredients that we're looking for. And in some ways, we make decisions the same ways as an investor would make, looking for companies to invest within. The acceptance rate for companies within Bakar Labs to date is overall, we've had well over 200 inquiries. And like I mentioned, we've had 43 companies within the incubator.
Oh, wow.
It's, greater than 90% of the companies that we've offered space to have accepted that offer. But, that said, if it's a compelling technology market fit with a great team, we really want to hear the story, and we really want to help accelerate that company.
Yeah, so how about like startups getting funding? Like, do you also accept, like, is funding a big part of the application process, like maybe pre-seed funding, or is it startups that may have no funding completely can also have a better chance of applying and also getting a space?
Yeah, I mean we're an incubator that offers lab space, office space, and meeting space, conference space to companies. We do charge rent. We charge fair market
Mhm.
rent rate for the East Bay, which we're required by law to. Um, since we're a not-for-profit entity, we don't have the ability to use our tax-free status to undercut the private market. So we have to compete with the private market at the same, at the same rates they charge. We can't undercut them. But so a company has to have enough capital to pay the rent for a reasonable amount of time. But we set that at a pretty low bar at around $250,000. That's the level typically of a phase one SBIR, small business
Mhm.
innovative research grant from the NIH or the NSF. So if somebody has a phase one SBIR or a small friends and family raise, capital's not a barrier for them to come into the
I
incubator.
see. But there has to be capital, right? It can't be like, oh, I have an idea, but I don't have a product and there's no funding yet, right? There has to be some funding, right? Not too much, at least.
Yeah, like I mentioned, we have a pipeline of programs that runs all the way from funding late stage academic research to all the way on the other end, a company getting a large Series B and outgrowing us and moving to their own space. So we have a couple of programs that support research that's right at the juncture of becoming a company. So I direct this program called the Baker Fellows Program, which provides $300,000 grants to faculty who are developing late stage academic
Mm,
research to get it to the point where it's shovel ready to become a company. We run a couple of mentorship programs that take nascent ideas and help build them into companies that can go do a successful pitch, a successful raise, and then turn into one of our tenant companies. And we also, UC Berkeley has a number of university affiliated VC funds, small VCs that are looking for investment opportunities. And they're the ones who at least participate in, or in some cases lead that initial investment that provides the company with the capital that enables them to come into Baker Labs.
yeah. So what advice did you receive during your career that shaped your professional development or success?
Um, Probably a couple of things. One is if I ever hit the point where I'm not on the steep part of a learning curve, I'm doing something wrong. I always, every day, every minute want to be learning something new. And if I feel like I've learned something pretty well, like I'm not bad at IP law, I'm not bad at contract law now because I went through experiences, then I'm going to learn something new. I'm going to do a deep dive into cancer immunotherapy or autoimmune disease. I always want to be learning. And probably the second piece of advice is that when I was, a lot earlier in my career, I hesitated I would hit the books, which is effective, it's an effective way to learn,
Mhm.
to go into literature. But I hesitated to ask friends and colleagues and new acquaintances to educate me, to ask them questions. And the reason I did that is because I was a young assistant professor, and I figured if I started asking people questions, at some point they may say, "Oh my gosh, this young person didn't know the answer to that question. Wow, I'm not sure if they should get tenure." That's an oversimplification, right? But it's a little bit, if you're at that young, vulnerable stage trying to develop a career and develop your reputation, it's hard to admit ignorance about a topic. And I found out that was totally wrong. People, especially my colleagues at Berkeley who want me to get tenure, wanted me to, um, are happy to share their knowledge. People like being asked things they know. They like teaching. They like, sharing their knowledge with younger people, especially. So as time has gone on, I still hit the books. I still dive into the literature, but I have a lot more conversations with people who are more experienced, more knowledgeable, have more perspective in an area than I do. And I'm likewise am much more likely, like I am today, to take the phone call and talk with people and try to kind of pay back into the system and share a thing or two that I may have learned.
Yeah. So just one more comment. I know you talked about how like you always want to learn. Is that why a lot of professors, when their company gets to IPO or maybe they get to like a later stage, they kind of not get involved in the company just because they want to learn something else? Is that a reason why?
It depends on the person. So I actually, I pulled away from, I mentioned 4D in 2022. so I made time for other companies. I made time for Bakar Labs because that's exactly the time I was taking on Bakar Labs. And also because the company was really focused on clinical operations, and that's definitely not my specialty. But I stayed with the company for a number of years, including well over a year after we took it public. I myself, the CEO and the chairman of the board, participated in the IPO. It was a very well subscribed IPO.
You
We
cut the
upsized.
ribbon?
I'm sorry.
Do you, so people go to I think New York and they cut a ribbon or something.
Oh, they hit the bell at the
They
NASDAQ to
hit the
close.
bell, they cut the
Yeah.
ribbon. I
It
was
was in the middle of
wondering.
COVID, so it was kind of an odd
Oh,
time.
oh,
Um,
okay.
but I did the 75 investor meetings over 2 months because I wanted to learn. I was on the board of directors of the company because I wanted to learn and wanted to participate. And then new opportunities come up to learn, and so I pivoted to earlier stage things. But learning a ton along the way, and as long as I'm continuously learning, I'm doing in my own book, I'm doing something right.
So do you have any other comments that you want to talk about?
What are your interests? What is it that interested you in helping other people spread their knowledge?
Well, I feel like biotech is to the point where it's like it's a good industry, but it's not like as widespread as tech companies. You know what I'm saying? Like I feel like tech is they're a lot more open. Like if you look at the founders, whether it's Mark Zuckerberg or more controversial Elon Musk and something like they're more open. See, so people get excited about all these technologies, right? But I think biotech, like there are also a lot of exciting technologies, but a lot of people commonly ignore them because they might think, oh, like there's not much traction, and they see like Mark Zuckerberg getting traction, so they all go towards. So that was the main purpose of the show.
Yeah, it's interesting. This is utter speculation that the time to market for tech is a lot quicker than biotech, right? Because in
Yeah,
biotech, you have to invent the technology, you have to show it's efficacious and safe in animals. You need to run a 7 to 10 year human clinical trial process to get it approved, and then at that stage, it starts being available to the public. Whereas in tech, if you identify a market need for an important piece of software, an app, a hardware company like Tesla is different, but for a software company, you can go to market relatively quickly. And so I think it ends up being the case that a single personality can be at the helm during that entire process. Whereas in biotech, typically the company is handed off. The management evolves throughout that entire time. So I think in tech, the new products become more readily identified with a given personality, whether it be
yeah, that's
Steve
true.
Jobs or um, Bill Gates or Elon Musk or others. Whereas in biotech, it takes a huge village in order to get that early stage technology all the way from discovery to commercialization. And
Yeah, I
so,
mean, yeah, man, my comment was like there isn't this one person, like a one guy that people can identify as like, oh, that guy is like the biotech guy. He runs everything, like there isn't that one person. Like I know people that are really close. I think Bob Langer is someone that could be the biotech guy, but he isn't very public as a person. So I mean, it's also kind of like you need to find like the right personality and person that could kind of be open. You know what I'm saying?
yeah, I can't speak to Bob's interactions with the
Yeah,
public as a whole. Within our field, he's incredibly accessible.
yeah.
So he's
Like yeah,
a terrific citizen. Yeah.
yeah.
Uh,
So yeah, I would actually agree with that. He went on, his son went on. So there we go.
mhm.
All right. So thank you, Dr. Shade, for being part of this episode. And so people can get to know more about your insights into gene therapy and also business.
My pleasure. Thanks so much for your terrific questions and also for your initiative and creativity in creating a series like this that I very much look forward to continuing to listen to.
Mhm.